Okay. So we got isolation of an aerobic nitrogen fixing microorganism from soil. So nitrogen fixing now. So the background's green. I don't really know why people associate green with nitrogen, but they do nonetheless. Okay. So last lab we talked about the reduction of nitrogen and how it can go away from the soil, right? So it's reduced down to a gas, and then it goes away. They can leave that soil as a gas. So this lab we're going to talk about how it's replenished, and how. And so we also talk about how if there's no nitrogen in the soil, then there can be no growth of the crops, right? And so we have to replenish this nitrogen, and so we have to get microbes to fix nitrogen into the soil, and that way we can use it to make amino acids, nitrogenous bases, phospholipids, whatever it may be. And so again, we talked about how that's the reason why you can't constantly grow on crop fields. You need to give it a chance to rest to get the crop yields that you are most optimal. Right? So you don't want to run out of nitrogen. You need to replace this. So the way that we add nitrogen to the soil is by taking nitrogen that's in the air, that N2, and fixing it as ammonia. All right, and we'll talk about that. So what happens is that you can have a symbiotic relationship. So some prokaryotes can fix nitrogen, but they have to be in a symbiotic relationship with legumes. Okay, I think experiment like 34, 35, 30, no, it's 36. Okay, so yeah, so the experiment 36, if we flip to it, should be on page 197. Talks about the symbiosis, the rhizobium legume mutualism. I think that has to do with nitrogen fixation. Yeah, it does. It does. Yeah, so that it kind of goes over more there, but some of them have to be in that, relationship to fix nitrogen. Like rhizodium lives on the root nodules of certain legumes. But you can fix it without being in the symbiotic relationship. So there are free-living nitrogen fixators. And they occur both anaerobic and aerobic, and they also can occur up to the thermophilic temperatures. So they're necessary, and it's good that they can do them under a whole bunch of different conditions. So the process of nitrogen fixation occurs because of the nitrogenase enzyme complex. All right, and what this complex is, it's two enzymes are required. Those two enzymes are dinitrogenase and dinitrogenase reductase. Okay. And what these two enzymes do is they convert the N2 into ammonia. So the nitrogen gas into ammonia. And then this ammonia can then be converted into organic nitrogen by incorporating into glutamate, which is an amino acid. And the you incorporate it into glutamate, and it's really interesting, that whole process of nitrogen fixation. But the problem is that this enzyme is deactivated by oxygen, so you have to protect it. And so there's a whole bunch of different ways that you can do this. Some use oxygen so quickly that it doesn't... Sorry, hold on one second. Okay, I'm all good now. So cyanobacteria, so yeah, you can just use up the oxygen really quickly, so it doesn't affect the complex, right? Cyanobacteria, what they do is they compartmentalize their nitrogenase, so they put it in these like nice little vesicles called heterocysts, and then they do all the reduction in those vesicles, but those vesicles don't allow in oxygen, right? Azotobacter, what it does is it uses a third protein, and what that does is so you can think of oxygen as like an inhibitor. Right, it is an inhibitor that it attacks either at the active site or at an allosteric site, whatever it may be, but it has to attach to that enzyme in some manner to change the conformation of it. So what Azotobacter does is really smart. It uses a protein to prevent oxygen from attaching to the complex. And so because the oxygen can attach, it cannot deactivate, and so this third protein just protects the deactivation of the conformation of oxygen, right? So it's like it kills the inhibitor, so it prevents the inhibitor from binding, rather. So it doesn't affect the complex. And then also on top of that, Azotobacter also has this capsule, which doesn't allow things to move in and out as easily as possible. So the capsule kind of protects it in that manner too. So it's kind of got a twofold little protection scheme. And so what we did in this experiment was that we had a nitrogen-free broth. All right, and so it had no nitrogen in it. If you don't have nitrogen, nothing can grow, right? You need nitrogen in a lot of different areas. If you don't have nitrogen, you can't grow. So it's like, it's good with that. All right, and so what we did was we then put soil into it. So yeah, there's going to be a little bit of nitrogen in that soil, so yeah, you can kind of grow some stuff maybe. But theoretically, at least, only nitrogen fixers can grow inside of that broth, right? So we put the soil in and we grow some, but there are going to be some other things growing, and we want to make sure we're all good. So, so pause. It also has mannitol in there, which is a carbohydrate that favors Azotobacter growth. But so we're going to put the dirt in that broth, and we're going to let it grow overnight. We're then going to take so most of the things that do nitrogen fixation are like use oxygen to grow, and so a lot of the bacteria that we want to use are just on the top. So you want to make sure not to like shake it up and then do that stuff. But you want to just take a little loop full on the top of your broth that has the dirt in it, and you want to streak it onto a nitrogen-free plate. All right, and what that does is it further selects for Azotobacter, so it has mannitol in it, but it then also allows just the growth of, again, microbes that have to be nitrogen fixers. So what we then did was we grew that overnight, and then we took a plate of those and we did a gram stain, a capsule stain so that's it. So we just did a gram stain and a capsule stain of the bacteria, and a lot of us saw a capsule on it. So that's good because Azotobacter, this is what we've been selecting for, and Azotobacter has capsules, so that's good. Gram stain, I think Azotobacter is let's see. Azotobacter, I forget what Azotobacter is, but it's got the gram shade should work as well. Sorry. But this is a really good example of using both morphology and physiology to identify bacteria, because you're using the physiology of growing in a nitrogen-free plate, right? Having those enzymes that can fix nitrogen, that's its physiology. And then once we know that we limited it down to things that can fix nitrogen, we could then look at it and say, okay, so we know that there's a lot of things that can fix nitrogen, but look, this one has a capsule on it, so it's probably Azotobacter. We still need to do further tests to just confirm that, but it's probably going to be Azotobacter. And so that's a cool way of kind of like incorporating both the physiology and the morphology. So the takeaway here is that bacteria can recycle nitrogen by fixing it and to allow like plants to grow and stuff like that. Nitrogen fixation is important more than just growing plants, right? But that's just an easy thing that we can identify with. These bacteria can then be isolated and using a nitrogen-free broth and a nitrogen-free plate. And then we can use the morphology and the physiology to then identify the bacteria. So it's a really interesting experiment. These nitrogen experiments are some of my favorite ones that we do. So they're fun. I enjoyed them. They're cool. But that's it for the nitrogen experiments. A little 9-minute video. That's it.